Okay, and now let's proceed to the other species of mycobacteria, no, other than Mycobacterium tuberculosis. So let's start with the Mycobacterium bovis. Mycobacterium bovis has been a big help, no, in the healthcare industry, wherein this is used as the Bacillus calmette-guerin, or the BCG vaccine, which is prepared from a strain of the attenuated virulence reduced live bovine tuberculosis bacillus, which is the Mycobacterium bovis. Again, Mycobacterium bovis is a bovine tuberculosis bacillus, in which ang Mycobacterium bovis, no, na-lose niya yung kanyang ability to cause disease in humans. So that's why it is used as a vaccine. This Mycobacterium bovis grows very slowly on egg-based media. They produce small, granular, rounded, non-pigmented colonies with irregular margins after 21 days of incubation at 37 degrees Celsius. Okay, sa Middlebrook 7H10 medium, these colonies are similar to those of Mycobacterium tuberculosis, but they are slower, okay, to mature compared with the Mycobacterium tuberculosis. Now, Mycobacterium bovis, they produce TB, no, among the cattle, sa mga animals, and other ruminants, as well as mga dogs, cats, swine, parrots, and even in humans. Okay? Most strains of Mycobacterium bovis are niacin negative. Okay? They do not produce nitrate, and they do not grow the um, uh, in the presence of nitrate. All right. Okay. So now there is a classification, no, of the mycobacteria other than tuberculosis with the use of the Runyon's classification. Okay, wherein the organisms, okay, are being classified according to their pigment production. Okay, so now let's start with the classification ng Runyon, which is the photochromogens. Photochromogens are group 1 mycobacteria. They produce yellow to orange pigmented colonies that are only, no, they are only produced, no, when once they are exposed to light. From the name itself, photochromogens, they produce colors or pigments once exposed to light. Okay, for example, the Mycobacterium kansasii, okay, they produce yellow color. That's why they are known as the yellow bacillus. Their colonies appear smooth to rough with wavy edges and dark centers when grown in Middlebrook 7H10 agar. Now, the Mycobacterium marinum, okay, marinum, this means of the sea from the name marinum. Okay, so this is the Mycobacterium agent of the swimming pool granuloma. Okay, this has been implicated in the diseases of fish and isolated from the aquariums. All right. Now, the Mycobacterium simiae, wala siya sa slide. Okay, Mycobacterium simiae, SIMIAE, were isolated from the lymph nodes of the monkeys. Simiae, di ba? Simians, mga monkeys. Okay, so Mycobacterium simiae were isolated from the lymph nodes of the monkeys. Okay. Now, another classification according to Runyon, okay, the scotochromogens, which are the group 2. Okay, scotochromogens. So these organisms produce the color yellow to orange even without the light. Okay, so scotochromogens, they produce colors without the presence of light or exposure of light. Okay, so they're pigment, no, in light and darkness. All right. Now the Mycobacterium sanopi. Okay, this is the first isolated mycobacterium from the African toad. Okay, good to know. No, the Sinopi or Kerokeropi, Sinopi. Okay, first isolated from the African toad. So this organism has branching colonies with aerial hyphae, no, on the cornmeal agar, which is described as the bird's nest. So it looks like a bird's nest, no, branching ang kanyang colonies, may aerial hyphae siya. Okay, so wala siya bird's nest. Imagine ang bird's nest, di ba? Okay. Now the Mycobacterium gordonei, okay, which is known as the tap water bacillus. Okay, this is a tap water bacillus. All right. Another Runyan's classification for your mycobacteria is the non-photochromogenic, or the group 3. Okay, they produce non-pigmented colonies in light and darkness. From the name itself, non-photo, they are non-pigmented colonies, okay, in light and darkness. So example of your non-photochromogenic na mga bacteria, mycobacteria, are your Mycobacterium avium, no, Mycobacterium intracellulare. Okay, Mycobacterium avium and Mycobacterium intracellulare. They are part of the Mycobacterium avium complex, or the MAC. Okay, again, Mycobacterium avium and Mycobacterium intracellulare are part of the MAC, the Mycobacterium avium complex. These are known as the battery bacillus. Okay, so these are the battery bacillus. Okay, so this complex, no, these are the opportunistic pathogens sa patients with AIDS and other immune dysfunctions. Okay, now Mycobacterium terrae, no, also known as the radish bacillus. Okay, now Mycobacterium terrae is the radish bacillus. Okay, now another Runyan's classification is the rapid growers, or the group 4 mycobacterium. They produce visible growth within less than the 7 days. Okay, so they are rapid growers. Okay, so examples, si Mycobacterium fortuitum chelonae complex, which grows in McConkey, modified McConkey, okay, without the crystal violet. All right, Mycobacterium fortuitum, this is a group wherein it is the most common rapidly growing mycobacteria associated with localized cutaneous infections. Okay. Now, another organism that is of the clinical significant, no, among the humans is the Mycobacterium leprae. So this is the causative agent of the Hansen disease, or also known as the leprosy. Okay. This is an infection of the skin, the mucous membranes, and the peripheral nerves. Okay, so take note, no, Hansen disease is not considered highly contagious. Okay, and the Hansen disease, or the leprosy, has two major forms. We have the tuberculoid leprosy and the lepromatous leprosy. Okay, when we say tuberculoid leprosy, this is a leprosy that has restricted pathogenic growth, and it has a relatively high cell-mediated immunity. Okay, so involve ang ating mga macrophages, ating neutrophils, mga T lymphocytes, and other WBCs. It has a less severe case of leprosy, and it doesn't involve a higher amount of bacterial load when you say tuberculoid leprosy. Okay, the symptoms, no, include skin lesions and nerve involvement, wherein there's loss of sensation na kapag may nerve involvement na. Okay, patients eventually exhibit an effective cell-mediated immune response. All right, but when we say lepromatous leprosy, no, this is extensively spread all over the body. Okay, it has a poor cell-mediated immunity, so hindi nagte-take function ang ating mga cells, no, such as the macrophages, yung mga WBCs natin. The most severe stage of leprosy is the lepromatous stage. It involves a relatively higher bacterial load. Okay, so ang lepromatous leprosy, no, may skin lesions siya, and the lesion is progressive. Then symmetric nerve damage also occurs, and it does not produce an effective cellular mediated immunity response. So kawawa ang patient with the lepromatous leprosy. Okay, now how to diagnose your Mycobacterium leprae? Now these are found in parallel or palisade arrangement, which appears as cigar packets. Kapag cigar packets, gani, it's your Mycobacterium leprae. Again, kapag cigar packets, these are your Mycobacterium leprae. Okay, palisade siya in groups, but parallel, no, and described as cigar packets. So they contain this phenolase, okay, which is obtained from lepromatous skin nodules, which differentiates it from the Mycobacteria. Okay, Mycobacteria or other Mycobacteria species. Now, as what I've said earlier, Mycobacterium leprae cannot be grown in vitro. No, they are non-culturable on artificial medium. They can be grown experimentally only in the footpad of the mice. No, success is in the low temperature at 30 degrees Celsius of their footpads. No, and in the laboratory, known ang armadillos. No, these are the animals with low body temperature. Okay, so kaya sila pwede sila makuha doon or ma-isolate doon yung ating Mycobacterium leprae. Okay? And so, how to diagnose or how to detect this Mycobacterium leprae, no, in humans via the lepromin test. Okay, this is a skin test which consists of a heat-killed suspension of Mycobacterium leprae. These are prepared from lepromatous nodules, and this test, however, lacks specificity. So, dili siya specific with Mycobacterium leprae. So, what are the reactions of the lepromin test? No, early Fernandez reaction, wherein the nodule appears in 24 to 48 hours. And also, we also have this type of reaction, the late Mitsuda reaction, wherein the nodule develops in 3 to 4 weeks. Okay, so dugay-dugay pa yun, 3 to 4 weeks. And again, no, this lepromin test has a lower, or it lacks no specificity for the Mycobacterium leprae. Okay, and so that's it. No, that's it. That's all about our topic on the Mycobacterium. I hope you have learned a lot, and I would like to remind everyone to kindly read the book, okay, to further enhance your knowledge, to further grow your knowledge about these Mycobacterium organisms. So I recommend The Lost, not The Lost book, na the author of the Introduction to Diagnostic Microbiology for the Laboratory Sciences, no ba? Okay, and also Mahon, and also Bailey and Scott's, no? But The Lost has the most simplified, no, na discussion about mycobacteria. So that's it, everyone. God bless on your examinations, and good night.